So, you've just found out your child has Fragile X syndrome. I know you must be confused, concerned, worried, and I know you must have a million questions. But it's okay. I'm here to help you learn a little about your son's disease today. I'm going to tell you how it is caused and how he has inherited it, and hopefully you guys will leave here feeling more comfortable and more knowledgeable on his condition, and you guys will feel a little bit more at ease once you are more informed and know all about it. First, I will give you a small overview of Fragile X syndrome itself. Fragile X syndrome is a genetic condition that results in developmental problems like learning disabilities and cognitive impairment. It can occur in both genders, male and female, but it is more prominent in males, and it can result in mild to severe intellectual disabilities. Next, we will talk about some of the physical features and signs associated with Fragile X syndrome that you may already see in your son or you may see later down the road. First, in the physical features that are very common, you have a prominent broad forehead as seen on the figure here, large ears, a long face, a prominent jaw. You have a hollow chest, things such as scoliosis, and as far as symptoms go, there are autism spectrum disorders, intellectual disability, like I had said before, and very distinct facial features, like you can see that listed. We will now move into the discussion of how Fragile X syndrome is caused. Fragile X syndrome is caused by changes in a gene called the Fragile X mental retardation 1 gene, otherwise known as FMR1. Now I will go into a little background on the FMR1 gene. The FMR1 gene is located on the X chromosome. The FMR1 gene codes for the Fragile X messenger ribonucleoprotein, otherwise known as FMRP. And FMRP is very essential because it supports normal cognitive development. And on the FMR1 gene, if there is enough mutation to where there's a high enough number of trinucleotide repeats, it will result in having Fragile X syndrome. So, how does your child inherit Fragile X syndrome? Well, like I had mentioned previously, Fragile X syndrome is a genetic disorder. So in order for a child to inherit Fragile X, one of their parents must carry the mutation to pass down to them. And a process called genetic anticipation makes it more likely for the next generation to exhibit Fragile X syndrome once the mutation is passed down from generation to generation. I will now elaborate on the process that I just mentioned called genetic anticipation. Genetic anticipation is a process in which signs and symptoms of genetic conditions become more severe and or passed down at an earlier age due to the fact that they are passed from one generation to the next. In the case of Fragile X syndrome, an increase in trinucleotide repeats passed down from one generation to the next is the major factor in exhibiting the signs and symptoms of Fragile X syndrome. Here, I have a pedigree illustration to help you better understand how Fragile X syndrome is passed on from the parents to the next generation. Here in this illustration, squares will represent the father, or males. The circle will represent females. The ones shaded in black will represent the abnormal FMR1 gene, and the ones that are white will represent the normal FMR1 gene. In the first example here, it shows a father with mutated gene and how his offspring will turn out. As you can see, the 2 males are normal and the 2 females have inherited the mutation. This is because a father can pass down his X chromosome to his daughters, but he can only pass down his Y chromosome to his sons, meaning that the father cannot give a son Fragile X syndrome. In the next example here, it shows a mother with the mutated gene. As you can see in her offspring, 50% of her children inherited the mutation for the abnormal FMR1 gene, and half of them did not, and they ended up normal. So in your case with your child, you, the mother, had to have been the one with the mutated gene, since you have a son who has Fragile X syndrome, because of the father not being able to pass Fragile X syndrome down to his son. Here I have another example of an illustration that explains the inheritance of Fragile X syndrome. And here, this one, the white letter will represent the typical FMR1 gene, which is the normal, and the shade in black will represent the Fragile X mutation, either whether it be a pre-mutation or full mutation, which we will get into what that means later. And then the Y is obviously the Y chromosome, which will never be shaded in black because FMR1 gene is only on the X chromosome. So here in this example over here, we have a father who is a pre-mutation carrier for Fragile X, and then a mother who is typical, she's completely normal. And they have 4 kids in their offspring, 2 daughters and 1 son. So as you can see in the result here, and as the lines are drawn, both of the daughters had to inherit Fragile X because they have to receive an X chromosome from their father, in which he only has 1. And with the sons, they are of course both normal because they have to receive their X chromosome from their mothers and the Y from their father, so they were not at risk for getting Fragile X. In the second example over here, we have a mother that's a pre-mutation carrier for Fragile X syndrome, and we have a father who is typical and is completely normal. They also have 4 kids, like the previous example, 2 sons and 2 daughters. In this example, 1 daughter and 1 son were typical and were completely normal, while the other daughter and son both received the mutation from their mothers. When the mother is a pre-mutation carrier, it is always a 50-50 chance whether they will pass it on to their children because of them having 2 X chromosomes. Because the sons will always inherit an X from their mother, which could be either one. As you can see, the son who did get the pre-mutation got the X that had the FMR1 gene that was abnormal, while the other son happened to inherit the X that was normal. And it's the same with the daughter, as they will inherit 1 X from the mother and 1 from the father, and it is also a 50-50 split on which chromosome of the X that they're going to inherit from the mom, whether it be the normal or the mutated one. As you can see in the illustrations I just showed, a mother is only able to pass on 1 of her X chromosomes to an offspring. This is because of a process called X chromosome inactivation. This is to ensure that, like males, females only have 1 functional copy of the X chromosome. And one of the X chromosomes that a woman possesses is turned into a bar body, which is an inactive X chromosome, so that they both cannot get passed on and only 1 can. We will now dive into what you've heard me talk about previously during this presentation of what causes Fragile X syndrome, being trinucleotide repeats. Trinucleotide repeats are a subset of a larger class of unstable microsatellite repeats that occur throughout all genomes, and in Fragile X, a certain type of trinucleotide repeat, the CGG, is what causes the disease. To explain a little more on what those classes of trinucleotide repeats means when it comes to Fragile X syndrome, people that are normal, have a normal range of base pairs, do not have Fragile X syndrome, and they do not pass on a higher chance of giving their child Fragile X syndrome. People between the intermediate base pairs, they do not have Fragile X syndrome and are not at risk of giving their children Fragile X syndrome, but can have children with a pre-mutation. And for people between the pre-mutation range, they do not have Fragile X syndrome themselves, they don't exhibit it, but they can have children with either a pre-mutation or a full mutation. And then people with full mutation of Fragile X syndrome. These are the people who do have Fragile X syndrome and exhibit the symptoms of Fragile X syndrome. And women have a 50% chance of passing it on to their children. So in Fragile X syndrome, Fragile X syndrome, people that are normal and don't have Fragile X syndrome have less than 45 repeats of the CGG trinucleotide. People who are considered to be intermediate cases have between 45 and 54 repeats of the CGG. People who are considered to have the pre-mutation have between 55 and 200 repeats of the CGG nucleotide. And people who have full mutation have over 200 CGG trinucleotide repeats. For further understanding on Fragile X syndrome, we have to talk about the processes of transcription and translation. Transcription is the process of going from DNA to messenger RNA, while translation is the process of using RNA template strands to create proteins. Here I have a visual for how transcription and translation work. Here, you see we start with transcription, which takes a strand of DNA and uses it to make a complementary strand of messenger RNA, which then in translation is used as a template strand to be converted into polypeptides. An illustration for how transcription and translation occurs in Fragile X syndrome. Here, for people with normal cases with less than 45 repeats, transcription and translation function normally, and they both occur, which results in the normal amount of FMRP, which again is essential for normal cognitive development. For pre-mutation cases here, with people between 55 and 200 repeats, you can see that their repeat section here on the gene is widened, and it results in still transcription and translation occurring, but less levels of it, and therefore less levels of FMRP are being produced. And here in the full mutation, people over 200 repeats of the CGG, you can see that their repeat section is very elongated, and there's even breaks in it, which inhibits transcription and translation from taking place, which means no FMRP is being produced, and Fragile X syndrome is very present and exhibited. Another process we can talk about is DNA methylation, a process in which methyl groups are added to the DNA molecule. When located in gene promoter, DNA methylation represses gene expression by acting as a roadblock for RNA polymerase. RNA polymerase is a multi-unit enzyme that synthesizes RNA molecules from a template of DNA through transcription. I have a picture of RNA polymerase here, and as you can see, it's a little blurry, but it takes the DNA and makes an mRNA strand from it based on the DNA template. Picture here of how DNA methylation acts in Fragile X syndrome. So here, the first example is someone that is normal and does not have Fragile X. As you can see, they have a normal number of CGG repeats, and DNA methylation is not occurring here. So RNA polymerase is able to go through with transcription, which means it makes the messenger RNA strands, which then get translated into the FMRP like normal. And then here, an example of someone with full mutation, and DNA methylation is definitely active and present here, and it prevents RNA polymerase from transcribing the DNA into messenger RNA, and therefore it cannot result in making FMRP, which results in Fragile X syndrome. We'll begin to wrap up our discussion by talking about FMRP's impact on translation. FMRP binds to messenger RNA to regulate what transfer RNAs bind to the messenger RNA, influencing what proteins are synthesized in order to establish that normal cognitive development between the synapses. FMRP has the ability to repress proteins by binding to regions called UDR regions of mRNA to suppress or inhibit translation. And here you can see is an example of it binding to keep transfer RNAs from binding to. As we've established throughout this presentation, FMRP is absent in Fragile X syndrome. And because of this, FMRP cannot regulate proteins to not be expressed, and the absence of FMRP inhibition and translation leads to the presence of Fragile X related symptoms. I hope with this presentation that you all were able to learn a lot about your son's condition today and are able to know now how he got it and what caused it genetically. And I hope that this makes you guys feel more comfortable moving forward, and I wish you all the luck with you guys and how you were able to raise your son. Thank you.